Platelets are cell fragments that help to create blood clots whenever you have damage to a blood vessel. So you can see in our picture here that they are pretty small, and so there are these little purple dots all around here that are the platelets. And then in this picture here, you can see that we've got some red blood cells and platelets, and then you see this kind of fibrous webby stuff here. So this fibrous webby stuff comes from that fibrinogen, which is the plasma protein involved in blood clotting. And we'll talk about how that happens on the next several slides because we're going to talk about hemostasis. And hemostasis is just the process of forming a blood clot. So there are three main steps involved in hemostasis, and we'll talk about each one of these in more detail. If the body is not able to clot, or if the blood's not able to clot, then even a small cut or a small injury could lead to excessive blood loss, which is called hemorrhaging or hemorrhage. And so thankfully for blood clotting, if it's not too bad of a damage to the blood vessel, then the platelets and the fibrinogen can clog up the area to prevent excessive blood loss from happening. So first let's take a look at step one of hemostasis, which is vascular spasm. So the first thing that kind of triggers the onset of hemostasis is that a blood vessel is damaged in some way. So this could include you maybe cutting your finger and it causes bleeding, or if you get hit really hard and you get a bruise, that's another example of a blood vessel that may have been damaged in the process. So when a blood vessel is damaged, if it tears the blood vessel wall, then the components of the blood start leaking out of the blood into the extracellular fluid or the tissue fluid of the surrounding areas. So the first thing that happens is that the smooth muscle in the walls of the blood vessels starts to contract. And this contraction helps to kind of pinch off the damaged areas so that it limits how much of the component are leaking out of the blood vessel. So it's basically if you're decreasing the size of the conduit, then less stuff can move through it. So this is a very quick response in order to kind of decrease how much bleeding is happening from the damage. But it's not stopping the bleeding completely, but it's buying us some time so that your body can then make a blood clot. So the next step is platelet plug formation. So the damaged area of the blood vessel releases chemicals. As the platelets are activated by the chemicals released from the injury site and by contact with collagen. So collagen is part of the blood vessel wall, and when it gets exposed, it's kind of rough, kind of sticky. So the platelets are activated by those chemicals released from the injured cells, and it causes the platelets to become kind of spiky looking. So you can see that these are kind of spiky, whereas these platelets are kind of more smooth. When they become spiky, they stick to each other, and they also stick to that rough collagen area. Then when the platelets bind to that collagen area, they actually release more chemicals to activate and attract more platelets. So this is actually an example of a positive feedback loop because the platelets come in, start to plug up the area, and then they send out chemicals to bring even more platelets to the area and to activate those platelets to make them stick. Once the platelets move to the source of the chemical signals, they bind there and the platelet plug starts to grow. So once again, this is not a long-term clot, but it's helping to buy time as a clot is forming because the actual clot is formed in step 3. Step 3 is called coagulation, and the big difference between a platelet plug formation and coagulation is this plasma protein called fibrinogen. So during coagulation, fibrinogen is converted to fibrin, and fibrin is this kind of mesh-like protein substance here. And this fibrin traps those platelets that are here, and this forms a more kind of I don't want to say a permanent clot because the clot does eventually go away, but it's a longer term clot in order to prevent blood loss as the blood vessel is trying to repair itself. So that fibrin helps to hold those platelets in place, and it helps to trap even more platelets and red blood cells. And so this is a longer term solution to limit blood loss as the blood vessel wall is preparing itself. Now, as I mentioned, it's not a permanent solution because if it were permanent, then there would never be blood flow through this blood vessel again. But the good news is blood vessels can repair themselves, and when they do, this blood clot can be broken down to reestablish blood flow through the area. So this is just an overview of all those steps that we talked about on the previous slides. And in order for the clotting to occur, there's a cascade. These are all clotting factors that have to be activated in order for fibrinogen to be converted to fibrin in order to create a clot. Now, you don't need to memorize this, but a couple of things I want to point out is that the damage to the blood vessel wall can happen within the blood vessel or outside the blood vessel. Either way, as long as the blood vessel is damaged, then this clotting factor cascade is going to be activated. So when one clotting factor is activated, it activates another one, which activates the next one, and the next one, all the way until you end up with a blood clot. And two kind of nutrients that are needed for this to occur are going to be vitamin K and calcium. So one reason why you need vitamin K and calcium in your diet is so you can produce blood clots. Now, as I mentioned, clots don't stick around forever. They do break down once they're no longer needed. So we could maybe call this the fourth step of hemostasis, and that's fibrinolysis. And the suffix lysis means to break. So this is a step in which the fibrin is broken down. And so it degrades the clot after the blood vessel wall has been kind of fixed or repaired, and then normal blood flow can happen in that vessel after it's finished healing. Now, there are some substances that oppose coagulation, and these are called anticoagulants. Some are naturally occurring in the body, and others can be used pharmaceutically. So some, because just like everything else in our body, you want blood clotting to happen when you need it to happen, but you don't want it to happen when it's not needed. And so some people do have clotting disorders in which blood clots form when they're not supposed to, and that can actually cause problems because it can cut off blood flow to certain areas of the body. So basophils, so these are some of our white blood cells there, they can release something called heparin, and we can use heparin pharmaceutically as well. But this heparin helps to prevent blood clotting from happening. So it helps to kind of balance out, to keep blood clots from happening when they shouldn't, but blood clots should still happen when they need to. We can also use anticoagulants pharmaceutically. So aspirin is kind of just a really well-known over-the-counter one. As people get older, it's recommended that they take a low dose of aspirin every day, and that just helps to prevent blood clots in people that may be at a higher risk for things like strokes and heart attacks. Now, there is a genetic disorder called hemophilia that affects the formation of clotting factors. So the platelets are fine, the fibrin is fine, but the clotting factors necessary to activate that are not there. And it might be a problem with only one clotting factor not being made. It could be several. But people with hemophilia have a reduced ability to clot their blood. So small injuries that someone with normal clotting abilities would survive without a problem could be fatal to someone with hemophilia. So for example, if I cut my finger, I can probably just put some pressure on it and a Band-Aid, and eventually my blood's going to clot and I'm not going to bleed to death. Someone with hemophilia, however, if they were to get cut, it could potentially lead to excessive bleeding that could cause them to die.